Well, welcome everyone. I'm Jen Farmer from the Friedrich's Ataxia Research Alliance, and we are in May, and May is FA Awareness Month. And this is our first webinar of the month with Lexeo Therapeutics. And I just wanted to make a quick announcement that we do have a special webinar series this month called our Flash Talks. And our first Flash Talk will be on Thursday, May 7th at noon. And if you haven't registered yet, I will put the link for our Flash Talks in the chat. Flash Talks are research updates provided by young investigators, grad students and postdocs, and they will give short presentations on the work that they're doing. So hopefully you'll be able to join us for all of our webinars this month in honor of FA Awareness Month. So today's webinar is with Lexeo Therapeutics, and the focus of the webinar is going to be an update on two clinical trials that were run, the Lexeo Sunrise study and a clinical trial at Weill Cornell with Dr. Ron Crystal. Both of these studies used the same gene therapy product, and the study designs were very similar. So Dr. Crystal is going to share with you the combined data from both of those studies. If you have questions as we go through the presentation, please feel free to put them in the Q&A box. And at the end of the presentations, we've allotted time for us to try and go through some of the questions that you may have. So with that, I'm going to turn it over to Nilomi Shah and from Lexeo Therapeutics and Dr. Ron Crystal. Nilomi and Dr. Crystal, thank you for joining us today.
Thank you. And thanks, Jen.
Thank
And
you,
thank you for everyone for joining, taking time out of your day to listen to this webinar. So I'm very excited to have Dr. Crystal and myself present some information that was actually recently accepted and presented as a late breaker at the American College of Cardiology conference. This is, as Jen mentioned, a combination of some work that Lexeo has done and Weill Cornell, looking at a gene therapy that is directed to treat the cardiomyopathy aspect of FA. Next slide, please. So this is just some disclosures for myself and Dr. Crystal. I am an employee of Lexeo Therapeutics. I work under the medical affairs team. Dr. Crystal is the professor and chairman of the Department of Genetic Medicine in Weill Cornell Medicine. He is also a part of Lexeo Therapeutics in terms of a consultant and a shareholder. And it's really exciting to have Dr. Crystal here because a lot of the early work about this program actually started in the Cornell lab. And so it's really great to see this program moving forward into the clinical trial results that we're going to be describing today. Just a quick note that the gene therapy that we'll be referring to today has not been approved by the FDA for wider use, so it's still being investigated in clinical trials. Next slide. Okay, so before Dr. Crystal goes into the study design and the results that have been shared to date across the two trials, I just wanted to take a step back and level set a little bit about how the heart is affected in FA. So when we talk about Friedreich's ataxia, what causes FA? Well, the ultimate cause of FA is a mutation or something that is incorrect in the frataxin gene or the FXN gene. And you could take a look at the left side of my slide here and I'll walk you through that. Because there is a mutation in the frataxin gene, it ultimately means that there is something wrong with the instruction manual that is led to write the code to develop the frataxin protein. The frataxin protein normally is really important in mitochondrial cells to help develop energy so that organs and other body parts can power and work effectively. So in FA, because there's a mutation in this frataxin gene, there's then lower levels of frataxin protein, and the organs that need the energy, such as the muscles, the brain, and the heart, are not able to be powered or work as effectively as they should. Why this is important is because when in FA, sometimes the most obvious signs and symptoms are the neuromuscular aspect of the disease. So this might be change in movement, unsteady gait, difficulty with coordination. But what's important to recognize as well is that the heart is also affected. And many times the heart is affected without any signs or symptoms being obvious. So it's sort of silently being affected. Um, Sorry, I'm hearing that there's a little bit of an echo. Can everyone hear me okay? Or maybe okay. No, okay, maybe it's just a couple of people. Okay, perfect. Thanks for confirming. Awesome. Okay. So, it's important to recognize that the heart is being affected, but it may not be as obvious or as visible in the beginning as some of the more neuromuscular or neuro aspects of the disease. But it is being affected because frataxin protein is needed in the heart. That's also why they say that, FA is a multi-system disease that requires a multidisciplinary care team, and a cardiologist is typically a part of that team or should be. It's also why when you look at the FA guidelines, they do recommend that you see a cardiologist once a year to understand what your heart health looks like. So, how is the heart exactly affected in FA? Well, without enough frataxin protein, like I said, the mitochondria in these heart cells are not able to work properly. They don't have the energy, and so the heart has to work harder than it normally does. That adds a lot of stress to the heart itself. And the more stress that's added, the harder it has to work, actually leads to the heart walls being thickened, which is something that you don't want. Many times you'll refer to the term hypertrophy. It's essentially, a term that is described when talking about the heart walls being thickened. And approximately 60% of individuals with FA can develop hypertrophy with the heart. Then over time, this leads to the heart, being stiff, less flexible. Think about your heart, it's pumping. And so if it's not flexible and if it's not accommodating the blood and being able to pump, it's going to make it difficult for the heart to pump that blood, pump it efficiently, etc. And so that is known as heart muscle disease or cardiomyopathy. So again, this might not be something you feel right away, but it's something to know that happens. And in fact, unfortunately, heart-related issues account for a majority of deaths or shortened lifespan in individuals living in FA. It's one of the reasons why both Cornell and Lexeo chose to focus on addressing cardiomyopathy and FA because it's such a significant outcome of the disease and an unmet need. Next slide, please. So, how does gene therapy exactly work? You'll hear Dr. Crystal refer to the gene therapy as AAVrh10 with the FXN. You may also hear the shorthand term that Lexeo specifically uses called LX2006, but it's the same gene therapy, so please don't get confused if you hear it described one way or the other. So, like I said, the cause of FA is a mutation or something not working properly in the code of the frataxin gene. So one way you fix something that's not working properly is you replace it with a copy that is working properly. So that's the idea of gene therapy. The idea is that you want to give the body a copy of the frataxin gene that does not have the mutation. It's working as it should, and by theory, if it works as it should, then it will have the right code to develop enough frataxin protein in the cells that need it. So in this case, the working copy of the frataxin gene is attached to or has a delivery vehicle called AAVrh10. So think of AAVrh10 as like a car with a antenna or a receptor. When it enters your body and with LX2006, it's given as just a one-time IV infusion that then goes into your bloodstream. So you might be asking, well, how does it know to go to the heart, right? So AAVrh10 is sort of that viral vector or that delivery that the frataxin gene is inside. And think of it as having sort of like an antenna that targets going to the heart, so the heart cells. It almost has an affinity or an attractiveness to go to the heart. It is when it's injected via IV, spread everywhere, but it has a specific attractiveness to go to the heart because of that antenna or that receptor. So the idea is you inject the product, the gene therapy product, one time through an IV infusion into the blood. It then travels through the bloodstream and goes to the heart cells, where it is released in the heart cells. Again, this is a working copy or a proper frataxin gene, which is then able to develop or write the code for frataxin protein, increases frataxin protein in those cells, and then allows the frataxin protein to do what it needs to do in the cells so that the heart cells can work properly and improve function. So it sometimes sounds like science fiction, but believe it or not, this has been tested in first in human clinical trials, one that was sponsored by Lexeo and one that was done at the Cornell hospital. And so what Dr. Crystal is going to now present to you is actually a combination of the results of those two trials. Again, it is the same gene therapy. The trials were developed very, very similarly with the same type of population, the same type of inclusion-exclusion criteria, very similar endpoints. And so what it allows us to do is to actually combine that data and look at it as one and provide even more evidence of where this gene therapy is working, where we're seeing, signals of benefit, and also most importantly, being able to follow the safety of this gene therapy. So I will stop there. And I will pass it over to Dr. Crystal to now take you through the results of those two clinical trials. Oh, Dr. Crystal, you're on mute.
No, I'm not. Thank you, Nilomi. And so let me go over with you all in terms of the trial design of the gene therapy for the cardiac manifestations of Friedreich's ataxia. As Nilomi, go back. Thank you. As Nilomi pointed out, this is a combination of two studies, one that we carried out at Weill Cornell, which was funded by the Heart, Lung, and Blood Institute at National Institutes of Health, where we studied 9 patients, and a very similar study that was carried out at Lexeo Therapeutics at several institutions around the country for 8 patients. These were open label trials. That is, we took patients as they came along. The virus containing the normal frataxin gene was administered intravenously at increasing doses, so 3 doses were used. And I'm going to show you the important parameters to assess the cardiac manifestations of Friedreich's ataxia, as well as the neurologic scale. So one is thickness, as Nilomi pointed out to you, that one of the problems that occurs in Friedreich's ataxia is the heart gets thicker as it's working harder and harder to try to pump even with less energy because of the deficiency of the frataxin gene. So one of the measurements we make is the cardiac MRI measurement of heart thickness, and this in the jargon is called left ventricular mass index, but it's basically the thickness of the heart. The second parameter that we look at are blood levels of troponin I. Troponin is a heart structural protein, and because the heart cells are working so hard to try to pump blood out of the heart, even in the context of not having enough energy, some of the heart cells get damaged, and some of the structural proteins of the heart cells leak out of the heart into the blood, and we can measure those. And this is called troponin I, and I'll show you that data. And finally, I'm sure many of you are familiar with the mFARS scale, which is a neurologic scale to assess Friedreich's ataxia specifically, and I'll show you some data with that. The combined study population were 17 individuals. There were 6 males and 11 females. The average age was 25. And as Nilomi pointed out, the frataxin gene has mutations or abnormalities in terms of its lettering, and these are called GA repeats, and the average GA repeats were 670 to 1176, so typical for Friedreich's ataxia. 11 of the patients had very early disease, so they had elevated serum troponin I levels, but normal thickness of the heart, so the very early in the pathogenesis of the disease. And 6 of the patients in the combined study had more advanced disease with elevated LVMI, that is thickness of the heart. And in both studies, there were periodic assessments, both pre and post therapy, of the thickness of the heart, the troponin levels in the blood, and the mFARS neurologic scale. Next panel. In terms of safety, intravenous administration of the virus containing the normal gene to the patients with Friedreich's ataxia was well tolerated. One of the things we worry about in gene therapy at very high doses is toxicity to the liver, or sometimes the immune system can be activated. But the doses that we're using here are moderate doses, and we saw no significant liver toxicity or immune system activation in either of the studies. There was a total of 4 serious adverse events, one possibly linked to gene therapy vector, although it's a little unclear because it occurred a year after administration of the drug. But there was inflammation of the virus, inflammation that might have been caused by the virus or could have been caused by the disease. That would be heart inflammation. It developed about 1 year post-therapy, and it resolved with anti-immune therapy. There were 3 other adverse events that were associated with immunosuppression. So in most gene therapy studies, we give immunosuppression to prevent any liver damage. And there were 2 related to infections and 1 to chest pain, all resolved and with no sequela. All other adverse events were transient, non-serious, or not treatment related. Next panel. So in the Lexeo study, several of the patients had cardiac biopsies by putting a catheter through the vein and taking a very tiny snippet of the wall of the heart. We could then, in the laboratory, could measure the frataxin levels. And what this shows is cardiac biopsy frataxin levels pre-therapy and 3 months following the therapy with the virus containing the normal gene. On the y-axis on the scale is the cardiac frataxin protein levels, and what is shown is the 3 dose groups. In the first dose group, there was 1 individual who had a biopsy, and there was a 29% increase in the frataxin levels in the heart. In the second dose group, there was an 81% increase, and in the third dose group, a 123% increase. So all patients with cardiac biopsies who had the therapy had increased frataxin levels above baseline. Next panel. This panel shows the thickness of the heart in all of the patients over time. And so this is the change in the left ventricular mass index, basically the thickness of the heart. And on the left-hand panel, which says pre-treatment, the shaded areas is normal levels, and the data is divided between females and males. And you can see some of the patients had thickness of the heart, and some of the patients had early disease and that did not yet have thickness of the heart that could be measured. But interestingly, as you see on the panel on the right, the individuals with the thickness of the heart that already occurred were in blue, and those who had normal thickness of the heart to start. And the data shown is percent change from the baseline. And so this is data in all 17 patients. And you can see every one of the patients either had stabilization of the thickness of the heart, or most had a decrease in the thickness of the heart. And interestingly, this was not only in individuals who had abnormal thickness of the heart, but also the individuals who had normal thickness of the heart, but were starting to increase the thickness and had suppression of the thickness as well. So a very, very encouraging result. At the last assessment compared to pre-therapy, 56% had decreased thickness of the heart, 44% had stabilized, and none had increased thickness of the heart. Next panel. This is the data on the troponin, the structural protein in the heart that comes from leakage of the heart as the heart is trying to pump harder and harder, and it gets damaged, and the cells leak out some of their structural proteins. On the left-hand panel is the troponin levels in the serum at start, that's before therapy. The shaded areas is the normal levels, and the dots are the levels of the patients, the females and the males. You can see almost all the patients had increased troponin levels in the serum. And on the panel on the right is what happened after therapy. A few of the patients had a little blip of increase, but all the patients had a decrease, a marked decrease in terms of the troponin levels in the heart, which suggests that the structural proteins are leaking out less and less because there's less damage to the heart. And even that one patient had an increase, looks like an increase, even that patient was within normal range. So very encouraging results. The thickness of the heart decreased, and the serum troponin I, the structural protein that leaks out of the heart, decreased. At the last assessment compared to pre-therapy, 94% of the treated individuals had serum troponin I that had decreased. Next panel. So this is an example of one patient. So many of you are aware of a parameter in the heart called ejection fraction. Ejection fraction is the fraction of blood that's pumped out of the heart compared to the amount of blood that's in the heart. And normally 50 to 70%, 75% of the blood gets ejected from the left side of the heart with each beat. If the ejection fraction is low, suggests that the heart is not working as well. And this one patient that I'm going to show you had a markedly decreased ejection fraction. So 16 over 17 patients had normal ejection fractions at baseline with no change with therapy. But this one individual had a low ejection fraction at baseline. And 18 months post-therapy, I'll show you the data, which is on the right. So in panel A is the serum troponin, the structural protein in the heart. And as you can see, the structural protein was very, very high in the blood at the start. But after the start, which is the vertical line, there was a marked decrease over time out to 18 months. Panel B is the thickness of the heart. And as you can see, before therapy, the thickness of the heart was very, very high. And after therapy, markedly decreased and stayed decreased over multiple evaluations over 18 months. And the panel C is the ejection fraction, the fraction of blood that's getting ejected from the heart with each beat. And as you can see, the ejection fraction of this one patient was 35% start, markedly decreased. After therapy, which is shown by the dashed line, there was an increase in ejection fraction. And over a period of time, it gradually increased. So it went into the shaded area, which is the normal range. And we actually saw this individual just recently and have another data point at 75%. So remarkable improvement shown. Troponin levels decreased, heart thickness decreased, and the ejection fraction markedly increased. Next panel. And finally, this is data in terms of the modified Friedreich ataxia, the mFARS rating scale. Although we directed the therapy to the heart, interestingly, we've seen some improvement neurologically as well, which may be skeletal muscle, maybe brain, or maybe a combination of both. So the mFARS, as many of you know, is a neurologic rating scale. The total scale score is 93. An increase indicates worsening, and a decrease indicates improvement. And it measures bulbar function, that is speech, limb coordination, and stability. And the natural history of untreated Friedreich's ataxia patients shows gradual worsening of the mFARS over time. But the data shown on the right, on the top panel, panel A, are the individuals in our trial that were taking Skyclarys that I'm sure many of you are aware of, which is an approved drug for the neurologic aspects of the disorder. And as you can see, for those taking Skyclarys, the dots or the red is when they were taking Skyclarys, is below the dotted line, suggesting the Skyclarys is having an improvement in terms of neurologic symptoms. But interestingly, we also had some individuals who are never taking Skyclarys, 8 individuals. And as you can see, except for that one individual, all the other individuals either stabilized or decreased in terms of their mFARS score, suggesting that there was improvement in neurologic function as well as cardiac function. Next panel. So in summary, gene therapy from the combined study of Lexeo and Cornell, gene therapy for Friedreich's ataxia with AAVrh10 frataxin showed that it's well tolerated. It reduces the thickness of the heart, reduces the levels of the troponin structural protein leaking out of the heart, and as I showed you, also has some neurologic improvement. And so our conclusion is More studies need to be done, but it may be efficacious for the treatment of Friedreich's ataxia. On the next panel, I just wanted to also mention that all the study that I reported to you, the combined Lexeo and the Weill Cornell study, were with adults 18 years or older, an average age of 25. But the Department of Genetic Medicine at Weill Cornell Medicine now shifted its focus to safety and efficacy of the same gene therapy in children ages 12 to 17 with early manifestations of cardiomyopathy. And we have approval from the Institutional Review Board and from the Food and Drug Administration. So we're starting the study. And if anybody's interested in that, contact Madeline Galbraith and her email shown there and her phone number. I'll leave it up for a moment so you can copy that if that's of interest of anyone in the audience.
Thank you all so much for participating today, all of you who joined and asked great questions. Dr. Crystal, Nilomi, thank you so much for sharing the results of these clinical studies with us. We will, like Nilomi said, we'll continue to make sure that we share information about the paper and the upcoming trials also through FARA's website and through our regular communications. And I hope to see you all on Thursday for our flash talks. So thank you again.
Much.
Thank you.
Great session.